The thing is that before we get on to the video, let me quickly tell everyone. So when you're coughing and you go to doctor's office, right? They take a swab out of out of your mouth. They take a swab, right? Every one must have gone through it when you're coughing or you have a lot of a cough in your bronchioles or throat. You go to the doctor. They take a swab. Now when they take a swab through that stick, they put that stick into a test tube. Now in that test tube they collect the swab from your mouth. So if you have a bacteria, then your test goes positive. If you don't have a bacteria, then your test goes negative, right? Streptococcus test done. So if you have a bacteria, your test becomes positive. If not, then it goes negative. So what exactly it is done is that swab is taken in a test tube. That test tube is sent to the lab. When it is sent to the lab, what is done is lab already has a nutrient media made. Okay, so the media is made on a Petri plate or flask. What they do is they take that wire loop. Okay, they take from the wire loop which I showed you. They put that wire loop in that test tube. They take the swab which was from your mouth. They put that on the Petri plate, evenly spread that on the Petri plate, and then put this Petri plate or flask for incubation. After 24 hours, if they see bacterial growth, that means you're infected. You had bacterial infection. If they don't see any bacterial growth, that means you didn't have the bacterial infection. In that also they will find out if the bacteria which is grown in that Petri plate, is it streptococcus. If it is like is it the bacteria causing the throat infection, they will find that out under the microscope. So what always they do is they first take the swab from the human beings or anybody, wherever they want to collect the bacterial culture. They'll collect it from that source. They'll bring it to the lab. Then what they will do is they'll make sure it is not contaminated. So what they will do is they will put this bacterial culture into a Petri plate. The Petri plate or the flask already has the broth or the agar, which has all the food the bacteria needs to grow. And then they will incubate and find out. So it's very simple. The Petri plate has the food or the nutrients which is required for the bacteria to grow. You put the culture using the loop, you put the culture on the Petri plate. You evenly spread it. If there is bacteria in the culture, in the liquid culture, that means you are infected. If there is no bacteria grown after 24 hours of incubation in the results, that means you are free of any kind of bacterial infection. And they will also find out under the microscope. They will, once they see those white color colonies are developed on the Petri plate, they will take those white color colonies with the loop, put it on the slide, put some stain, look it under the microscope. If they see the structure of streptococcus, that means you have streptococcus infection. This is in a nutshell how the research is done, right? So a bacterial nutrient media is prepared, then they put the sample on that media, they incubate it and check out if white color bacterial colonies are formed. If they are not formed, fine. If they are formed, they will take those colonies, put it on the Petri plate, on the slide, stain it and look it under microscope to see what kind of bacteria was present. Okay, now very important is what is this nutrients? What is that Petri plate or that broth I told you? This broth or the Petri plate has McConkey's agar. What is this? This is the food of bacteria. This food has all nutrients the bacteria needs to grow in the lab. So there is a specific oxygen, there is a specific temperature, there is a pH value of the liquid, liquid culture media. So all these conditions are necessary. If you don't have the right amount of pH, temperature, or the liquid culture media or the oxygen level, your bacteria is never going to grow. So it doesn't matter if somebody has an infection, but if your food which you're preparing in the Petri plate is not correct, your bacteria is never going to grow. You will get, the person might have a bacterial infection, but you will get false results. Why? Because bacteria will never grow. Why didn't the bacteria grow when the person had bacteria in his throat? Because your growth media or the food which you prepared in the Petri plate was not correct. So the bacteria never grew, right? Bacteria died, it never grew. Why the bacteria didn't grow? because the Petri plate didn't have the right amount of food. So to grow bacteria in a lab, you need to have the right amount of food or nutrients which are required for the bacteria to grow. So every bacteria has a different oxygen level, temperature, pH, and a liquid culture media. And there is a lot of research done hundreds and hundreds of years ago to make sure which bacteria needs what kind of pH or temperature. So based on that research, we make sure that when you make this food for bacteria or the nutrient for bacteria, that all those conditions are maintained. Okay, so everybody understood how in at surface level the research is done? It's clear? Yes. All right, so we will see a video now on how exactly it's done in the lab. Okay, so let me just pause this recording. So everybody understood, like looking at the video, how actually the research is done in the labs. When I used to be in the during my master's, during my university research days, this is how even any university you join, you will have to do research. When I was working many years ago in research labs, this is how the experiments are done. You first make the cultural media, the nutrients which is required for the bacteria to grow, and then you grow the bacteria on the take the culture, put it on the Petri plate which has the nutrients, incubate, and then you will see the bacterial culture for your research. Now, very important thing is, when I was showing you that bread thing, right, I was touching it with my hands. So you should use, basically you should always use gloves, like you saw in the video as well. You should use always gloves. So I washed, if you happen to touch something which has fungus, make sure you wash your hands with soap and hot water, or antibacterial solution and water. So that way no, it's all fine. Okay, so I didn't actually touch it, but I was touching the bread to show you guys white fungus. So it's always important to use gloves, and if not, at least because you have touched the bread, you make sure that you wash your hands. So for all of you, when you come outside from playing also, make sure you all wash your hands, and it's very important. I mean, and Nina later told me that I should have worn gloves, but I washed my hands with water and antibacterial solution. But make sure that we always when we are doing any experiments, it's better to always use gloves. Okay, so that's with microbiology. Anybody, if you want to do research, this is how the basic research and everything is done in the microbiology lab. If you guys want to cover, me to cover any topics which which are of your interest, please send me on WhatsApp or email me, or you can send through your mom's WhatsApp, that the WhatsApp Life is Kid Smile group, what kind of topics you want me to cover, and I will make sure I include those topics. Okay, so everyone have fun this weekend, and week, and we'll see you next time.